vesicular synapse, letter B, under Roman numeral 5. So we're going to look at a synapse, right? So this is going to be a communication point between one neuron and another neuron, or it could be between one neuron and a muscle, or one neuron and a gland. But in this case, we're just looking at two neurons here. So this is the axon. It's branching into the arborizations. We have our little boutons or terminals, and so this is what you see here, right? This is the terminal bouton. This whole thing is called the terminal bouton. And we can also call this the synaptic terminal. So remember that these guys come very close to, but do not physically touch the receiving cell. So the receiving cell is going to be called the postsynaptic dendrite, or in this case it's a dendrite, right, because it's a neuron. But it's going to be, we can talk about this membrane as the postsynaptic membrane. And then we can talk about this membrane here as the presynaptic axon, and the membrane being the presynaptic membrane. Okay, so presynaptic membrane, postsynaptic membrane, again, synaptic cleft or synaptic gap. We have our vesicles with our neurotransmitters stored inside. Remember that these neurotransmitters are actually created in the body of the cell. So if we go here in this image, here's our soma. We have neurotransmitters being cranked out with the ribosomes here, and they're literally just being moved, right, transported down these long axons, and then they get to the end where they're stored until they are released. So we can see the actual picture here. The cleft is very small, and this is where we would find our neurotransmitters being released. All right, so let's just take a larger look. This mitochondria are going to be the only organelles you find in the synaptic terminal because they create ATP, and it does take energy for these transport mechanisms, the exocytosis of the vesicle, to work. All right, so what we're going to look at is the terms. So we talked about number 1, the presynaptic membrane, and number 2, the postsynaptic membrane. The cleft or the gap, right? So let's talk about a couple things here. So we know that the neurotransmitters, when they're released into this cleft, there's going to be a receptor on the receiving end. Here, I'm going to draw here, right? So these are receptors that are going to bind to these neurotransmitters in the cleft. Now, when we talk about clearing the neurotransmitters, right? Because in order to stop the signal, if this dendrite, if the signal's electrical impulses stop, what is going to stop the signal from being passed on to the next cell is that we need to clear these neurotransmitters in the cleft. So there's 2 ways to do that. If you look at your notes, number 1, reuptake, and number 2, destruction by enzymes, you are familiar already with number 2 because we talked about, remember we drew a little Pac-Man in the cleft and we called that acetylcholinesterase. That's the name of the enzyme when you're talking about muscles, okay, the neuromuscular junction. But there can be various different kinds of enzymes here, and they all degrade the neurotransmitters in the cleft. But there's an alternative method, which is reuptake. So reuptake is going to be where you have a channel protein. Okay, so we're going to draw some channel proteins in the membrane here. These are also called transporters, and they're literally going to bind to the neurotransmitters in the cleft, and they're going to shoot them back up into the presynaptic cell here, right? So they're going to go back up in here. So they're recycling, right? They're taking these neurotransmitters and pumping them back into the presynaptic cell and clearing it, right? So there's 2 ways to clear. One with reuptake, and the other one is with destruction by enzymes. All right, so let's pause here a second. Talks about the action of neurotransmitters. So as long as you have these neurotransmitters in the cleft, that they're going to be active and they're going to be able to stimulate the receptors. Let me just draw back in the receptors here on the receiving postsynaptic membrane. Okay, but I want to talk about one interesting real life sort of use of this knowledge. There's a class of antidepressants called SSRIs. The most notable SSRI is Prozac, and the SSRI stands for selective serotonin reuptake inhibitor. So serotonin is secreted in the brain in areas where it enhances the mood, right? So if you have a lot of serotonin or active serotonin, adequate amounts, then you don't have a problem with depression. You have sort of quote unquote normal mood. But if people who are clinically depressed, one of the treatments is to allow serotonin to be more active in the cleft. And maybe the thought is that people who are clinically depressed may not be getting the amount of serotonin or the that stimulation from serotonin that they need. And so serotonin works on these reuptake mechanisms. So neurons that secrete serotonin will have reuptake transporters. And what Prozac does, right, it's going to selectively work on just the serotonin, just the reuptake inhibitors, right? So if you take Prozac is going to knock out these transporters. And so the serotonin in the cleft is not going to be able to go back into the presynaptic cell. Therefore, it's going to stay in the cleft longer, hitting these receptors more, and then you get the stimulation, right, that maybe you have more serotonin, or serotonin is just active longer in the cleft, and so it would help to enhance the mood or at least alleviate depression. Okay, so that's SSRI and Prozac, and so it's just a nice way of seeing a real-life application of this theory that you're getting in class. So let's just take a look at a picture I have here, right? So this is what I was drawing. So this is our serotonin that's being released into the cleft. And then these are the receptors on the receiving end. And then this is the transporter mechanism. So this is your SSRI, right? So SSRIs will block the uptake of serotonin. And this is not permanent, right? So Prozac, you have to take it every day, or you have to take, a certain dose every day, because these drugs will eventually, they kind of bind temporarily and then they fall off, right? So it's not a permanent binding. You're not completely destroying this reuptake, but it's going to bind for a little while and then pop off, but it slows down that reuptake. So let's talk about neurotransmitters for just a second. There are lots of different kinds of neurotransmitters in the body. Neurotransmitters are secreted all over the body, actually, not just in the brain, but also in the gut. and obviously to the muscles. So you have acetylcholine that is responsible for voluntary muscle contraction, and you guys, that should look familiar to you. There's other things called norepinephrine that's secreted in areas of the brain that make you feel awake. And then dopamine is one of those that you hear sometimes in the news. It is the neurotransmitter that's secreted in the brain in areas that really give you pleasure. So the pleasure center of the brain, that reward center of the brain, like if you get an A on the test, you feel amazing. If you go on a roller coaster ride and it thrills you, that's dopamine. If you you get some kind of gift that you love, that kind of like reward mechanism, you get a flood of dopamine anytime you're super happy. So this is also an addictive neurotransmitter. So people who are addicted to anything, it could be bungee jumping. You have those like adrenaline junkies out there that really need to have a lot of dopamine. And then you have people who just get a high out of gambling, or whatever the addiction is, they're seeking out that dopamine rush. So it is a problem in addiction. Serotonin we talked about with mood. It also has other jobs too, but you can see that there's different kinds of neurotransmitters. And one neuron makes one neurotransmitter. So you can say a neuron is GABAergic, which means it only makes GABA. A neuron is adrenergic, which means it only makes adrenaline or epinephrine. Cholinergic refers to acetylcholine. But the idea that I want you guys to remember is that neurotransmitters, when they are secreted into the cleft, they're going to be broken down either by reuptake or by the enzyme. And so they don't last a long time in that cleft. So in order to achieve a long-lasting effect, if you want the signal to keep going, like to feel happy or to keep the muscles contracting, then you have to continuously release the neurotransmitters into the cleft. Okay, so some interesting things here. This is just for fun. Psychoactive drugs and neurotransmitters. So drugs, and this doesn't have to be like illicit drugs. They could be prescription drugs. It could be natural sort of quote unquote drugs that we have in the environment, such as caffeine or in foods. Psychoactive just means that it's working on the brain, and it can make you feel different ways. And caffeine is a great example of this, because you know that caffeine is, seeked by people who want to feel more awake. They want to feel more alert because it has that effect on the brain. It's psychoactive. So it's a central nervous system stimulant. Caffeine activates the noradrenaline neurons, so that's that adrenaline, and seems to affect the local release of dopamine. So that caffeine high that you get, where you feel great, you want to get a lot of stuff done, everything's fine with the world, that's your dopamine talking. So what these psychoactive drugs do in terms of their effect on neurotransmitters is that sometimes they actually mimic, like the shape of the chemical of the drug looks just like the natural neurotransmitter that's in your mind, in your brain. And so marijuana and heroin are actually this way. They activate neurons because their chemical structure mimics that of a natural neurotransmitter in the body. Sometimes these psychoactive drugs can block reuptake transporters, just like the one I mentioned, the SSRI. And then some of them will cause more neurotransmitters to be released than normal. Okay, so like Adderall, for example, increases the activity of several neurotransmitters such as serotonin, norepinephrine, especially dopamine. So Adderall tends to sort of push out, right, more neurotransmitters than normal. Anyways, these are just some interesting ways to think about how your brain works. Your brain talks to each other, the neurons talk to each other through chemicals, and you can modulate, mediate, imitate these chemicals, and then you get these psychoactive effects, which is very interesting. All right, so let's talk about our last couple of things here, excitatory and inhibitory signals. So that's going to be this image. Let's take a look at this. So this is our neuron, which is naked. This is our neuron, which is more representative of what it is in your body. So you're going to have a single neuron, and you're going to have thousands, maybe, right, of vesicular synapses on the surface. So all of these blue buttons here, they represent the terminal boutons of incoming messages from different neurons. So you can see that one neuron can get messages from 1,000 different neurons. And so some of those messages are going to be called excitatory, and some of them are going to be inhibitory. So if you look at your outline in letter C, neurons can produce either inhibitory neurotransmitters or excitatory ones. Inhibitory means that when the neurotransmitter is released, oops, let me go to a picture of the neurotransmitter, right? So if this is an inhibitory neurotransmitter, when it binds to those receptors, it actually stops. It makes it more difficult for that postsynaptic cell to create an action potential, to do, to have an impulse. So inhibitory will make it more difficult to create an action potential on the receiving end, whereas excitatory, right, that's kind of what we've been talking about in class. When it binds, it creates an action potential on the other end. So there's both. And so that one neuron is receiving excitatory and inhibitory signals, and it's basically just a summation of what, is winning. Are there more excitatory signals? If so, it'll send an action potential. Are there more inhibitory signals coming? And that would be not sending an action potential. And I'll tell you that there's one disorder, Parkinson's disease, that there's a problem in the brain where the inhibitory signals are actually slowed down. Because the brain is not getting that inhibition, you have all these extra jerky movements of skeletal muscle. So the jerky, sort of uncontrollable muscle spasms that goes along with Parkinson's disease is because this area of the brain's missing its inhibitory, the quieting down of unnecessary muscle movement. So inhibitory signals are really important. All right, so that's it for that. Let's move on to our last topic, which is neuronal regeneration. Can neurons regenerate? So the answer is yes and no. A neuron here, if you damage a neuron too close to its body, no, it does not regenerate. Okay, the body is where everything happens, the nucleus, the organelles, and so no. And you know that neurons don't regenerate normally anyway. They don't undergo mitosis like the glial cells do. However, if you damage the axon of a neuron, so if the damage is farther away, farther distally away from the body, it can regenerate. So that is called Wallerian regeneration. It's pretty simple. There's damage. The myelin and all the axon distal from the damage is broken down, and it's degraded by your immune system, so it's just taken away. All right, so there's your macrophages. Those are the immune cells that are called big eaters, macrophage, right? And they're going to eat all that debris. Then Schwann cells will come in. Schwann cells are the heroes of the day, and they're going to come in. They're going to build a cord. They're going to build a cellular pathway that the axon will grow into. So it's going to follow the same path as the, what was damaged. And so the axon sends buds into a network of Schwann cells and then starts growing. And then you get this repair because of Schwann cells. So does this happen in the central nervous system or peripheral nervous system? So I'm asking you, where are Schwann cells? Are Schwann cells in the central nervous system or peripheral nervous system? You should answer peripheral. Okay, so this kind of fixing or regeneration of the nerve fiber, meaning axon, only happens in the peripheral nervous system. It does not happen in the central nervous system, the brain or spinal cord, because there are no Schwann cells. Schwann cells are the heroes. So let's say that you or someone there's an accident and they sort of cut a nerve, maybe say the ulnar nerve, right? That's actually your funny bone. So you have this deep laceration and you cut your nerve and it goes down from your elbow. So you lose sensation from your elbow down to your hand. You lose the function and you lose sensation. Well, you can actually get it back. So it takes time, but that nerve fiber, right, is going to grow very, very slowly and regenerate that nerve so you can eventually get sensation and movement back into your hand. And again, just to mention, we'll talk about this in chapter 14, but in the peripheral nervous system, the cell bodies are actually inside the spinal cord or they are very, very close to the spinal cord. And then it really is like what travels in your nerves is the long axon. Okay, so when you sever a nerve, if you have nerve damage in the extremities of your body, you're talking about you're damaging the axons. The bodies are safe and sound by the spinal cord, and that's why spinal cord injuries are so paralyzing, right? Because when it damages the spinal cord, you're damaging the soma of the neuron, and they don't regenerate, at least at this point, because obviously people are looking into their research and trying to get those to regenerate. All right, so which cells participate in the repair of damaged nerves in the PNS by forming a solid cellular cord that follows the path of the original axon? Hopefully this is very easy. Schwann cells. This question is a review question. Nerve impulses would travel most quickly through axons of which of the following characteristics? So this is asking you to remember the diameter of the axon is a small diameter, 0.5 micrometer, faster than a larger diameter, 20 micrometer. Well, your answer should be larger. The larger the diameter, the faster it goes. And then we want to look at myelinated versus unmyelinated. And hopefully this is easy. Myelinated, right? So answer B. All right, and that does it for this part.